Welcome to D.O.R.K.S. Good to see everyone today, and glad to have you here. I'm Scott Solomon, and I'm one of the co-hosts along with Kelly Weinersmith. And today our theme is communication. So we've got two great guests to talk with us today about their work and how it connects with the theme of communication. And so if you're new to D.O.R.K.S, let me just quickly give you the run through of how it's going to go. So in just a few moments, we will begin with our first presentation. After that, we will have an opportunity for a question and answer session. And we encourage you to submit any questions that you would like our first guest to answer, I should say. And you can do that with the Q&A button at the bottom of the screen. So the chat function won't work, but you can use the Q&A to submit questions. And then we'd be happy to ask as many of those questions to our guests as we have time for. Then we'll have a second presentation, and then we'll do the same thing after that second presentation. And then at the very end, we'll try to tie it all together and kind of bring it to a close. So again, welcome to D.O.R.K.S, and really glad you can be here. I do want to mention also, if this is your first time to D.O.R.K.S, we can there's a YouTube channel that we've got going now where you can watch all of the previous D.O.R.K.S presentations over the last few months. And so those are a lot of fun. And then we've got a Twitter account as well, which is at D.O.R.K.S Chat, where you can go to get information about upcoming talks and also find the YouTube link as well. So we always invite everyone, if they're interested, to share a drink with us as well. And so we've got a cocktail and a mocktail. And this week I'm drinking the cocktail. And this was an interesting one. We try to find something that will connect with our theme. And so this week, the cocktail is called the brain hemorrhage. And it's an interesting one. I have to say, it didn't work out quite as I had hoped for. The idea here is that it's supposed to look like a brain. And so it's got, basically it's got schnapps. The recipe calls for peach schnapps. I couldn't find peach schnapps, so I went with peppermint schnapps. And then it's got like Irish cream at the top. And because they're different densities, and because the cream is supposed to curdle in the schnapps, it's supposed to kind of look like a brain, but yeah, it didn't. It's separated, but it doesn't look very brain-like to me. And then there's a couple of drops of grenadine in there just to, I guess, make it look a little extra gory with some blood.
Could the switching of peach and peppermint schnapps have been the thing that caused the problem?
It might be. I couldn't, I didn't have the opportunity to get a peach schnapps, but it sort of seemed like it would be the same. Maybe there's something different between peach schnapps and peppermint schnapps. But anyway, let's see. It actually tastes pretty good. I mean, it tastes like peppermint candy.
That's good. That's better than brains.
I mean, yeah, as there were some comments on Twitter about how this drink might not look all that appetizing. Actually, it probably looks a little better than the photo did because the brain photo, it looked really cool, but it was maybe a little off-putting. Tastes great though. Kelly, what are you drinking?
So I'm drinking, so the mocktail is a peppermint pick-me-up, which is just coconut water, sparkling water, and a couple drops of peppermint oil. And it's quite refreshing. It doesn't look like a brain, which I consider to be a major perk. Even as someone who has worked with brains, I don't like thinking too much about brains while consuming beverages or food. So anyway, yeah, quite refreshing. And our buddy Steven, who's going to be our first speaker, has a drink with him too. What are you drinking today?
I'm a little boring and just went with what was in the house. Did not create any cocktails, so I just have a nice bourbon from St. Augustine Distillery down here in Florida. So quite tasty.
As like a smell and taste guy, is bourbon like popular in the community because it's particularly flavorful?
No, I mean, I think people have a lot of what goes on with your sense of smell and taste is your preference, what you've learned, what you've experienced, cultural. So people vary quite a bit, but certainly, yeah, whiskeys are particularly flavorful. They have a lot of volatiles. Bourbons have a little bit of a sweetness to them. So they certainly got it going on there.
Cool. And we also have our buddy Simon here today. Simon's drinking water, so he turned off his camera. We have no problem with drinking water. But Scott, is there anything else you wanted to say before I introduce Steven more formally?
No, let's go for it. I'm excited for these both these presentations today.
Okay, so Steven Munger is going to be our first speaker. He is talking about sniffing to learn odors as social cues. He's from the University of Florida Center for Smell and Taste. I actually met Steven while researching Soonish. We had a really weird question about mirror molecules. So the idea is that, what is it? Is a caraway and spearmint are mirror molecules. So they're sort of like your hands, like the molecules are just sort of flipped. And so we wanted to know if you had receptors that were mirror versions of themselves, would caraway taste like spearmint and would spearmint taste like caraway? Which was a very weird question. And you were very kind to us when we asked that question. And as I remember it, the answer was, that's a weird question, so no one knows the answer. Do we know? Have we figured it out in the last 6 years?
No, I mean, I think, and actually that interaction goes to one of the positive powers of Twitter, because I think that's how we ended up connecting, that the question is sort of put out there. No, I mean, you have hundreds of different receptors in your nose to recognize odors, and an individual chemical compound that is an odor, we call that an odorant, will activate more than one receptor, and each receptor will respond to more than one odorant, more than one chemical. So it's this sort of pattern recognition thing. So with probably a few exceptions, there isn't going to be a L-carvone and R-carvone, which are the two compounds for that odor of spearmint and peppermint. There aren't going to be just singular receptors that are different. They're probably going to be a combination of receptors that one of them does a little bit with the L with the left-handed, and one does a little bit with the right-handed version.
Got it. And I also remember that Zach and I had the question so poorly honed initially that we had even messed up whether we wanted to ask the question about smell or taste. And you were very polite in correcting us that we weren't even close on this weird question.
Well, no, it was actually, I don't remember that, but it's actually a very common confusion because of the way we use the word taste. It's so much we say, oh, this tastes bad, this tastes flat, this tastes great, whatever it is. When we say taste, we are usually not talking about it in the colloquial sense. We're usually not talking about the way taste and smell scientists would, which taste is exclusively the detection of non-volatile chemicals in the mouth that elicit sweet, salty, sour, bitter, or umami. Flavor is that combination of those tastes and smell. So it allows you to differentiate between a lemon and a lime, is because of the odor that goes along with it. So that lemon and lime is the flavor, the acid, the sourness is the taste. And so those words get conflated often.
And they certainly were conflated by us, but I think we added a footnote after talking to you about that to try to clarify it. But I guess not everybody has read our book, and so this mistake continues.
Well, those three people are now learning today. So
that's right. That's right. So you had a great fact. So your fact that you sent for the introduction was smell and taste researchers often have great food and drink experiences at conferences. And you shared some memorable ones. Salmon and aquavit under the bow of the Vasa in Stockholm. A variety of Belgian beers at every coffee break, reception, poster session, and dinner at Leuven. And fugu sashimi at Fukuoka. All right, so I butchered like 10 words in 2 sentences, but let's go with it. So what I want to know is, how did you feel when you found out you were about to eat fugu? Were you petrified? So can you tell people about fugu and your experience eating it?
Yeah, and I'm certainly no expert. So this was a small conference in Fukuoka, which is down in southern Japan, on Kyushu Island, and it was mostly a conference mostly with Japanese scientists, but a few from the U.S. and Europe. And so we came. There was a nice dinner from the host for those visiting speakers at the end, and had a variety of fantastic food. But at the end, and this was a restaurant where the chef was licensed to actually prepare fugu, which is a puffer fish that, for those of you who don't know, in their liver it makes a neurotoxin called tetrodotoxin, and if you have too much of this, it would kill you, which is generally a bad thing. But so in this case, it was just very thinly sliced sashimi, and so just the raw fish. It was very delicate. There actually wasn't a huge amount of flavor to it. It was sort of I almost equate it to eating slightly flavorful mini bubble wrap. It had sort of this poppiness to it in the way it was. I mean, it wasn't, hugely so, but a little bit. So it was interesting. It was pleasant. Yeah, it wasn't. I mean, you trust your hosts that they're not going to steer you wrong on this. But yeah, I mean, it's not something. It was a great experience, and it was an honor to be served that. So that was also part of it.
It surprises me to learn that the taste of fugu is not like mind-blowing. I can maybe understand risking your life for the best meal you've ever had, but for slightly salty bubble wrap, I'm not quite sure I'd put my life on the line for that.
Yeah, I may not be doing it justice with that description, but I think the reason it's so desired is because it does take a particular skill and training in order to do it. So it's, and I think that's true of a lot of experience of meals we have. You go to Alinea in Chicago, this 28-course tasting menu, where it's really art, it's entertainment. I mean, certainly I hope that the food is palatable and fun to taste, but it's an experience. And so meals are more than just ingesting the food and even just having that primal pleasure of what you're eating. It's the people around you, all of those types of things that make it. And so I think that's part of it.
Yeah, I think you're right. I ate moose heart once, and it definitely was all about the people and the experience, and not about the desire to eat moose heart.
That's one I have not tried moose heart yet. So
I've never tried fugu. I heard a lot of things about it. I think one of the things that I had heard about it was that it like really gives you like a tingling sensation that, from the tetrodotoxin that you're talking about, that's kind of part of the, perhaps enjoyable aspect of it. Did you
that wasn't the case here. And I'm not sure how much that is urban folklore type of thing, or if that's really a typical way. The story I've heard is that if you just slightly enough of the liver along with the flesh, that you would get that tiny buzz. I don't know if it's ever actually served that way or not, but it wasn't in this case.
Interesting. Yeah.
All right. Well, and the final fun fact is that Steven and I are neighbors separated by 40 years. We lived in the same neighborhood for a little while. We discovered that after I sent a package to Steven. So we have both spent some time in Charlottesville and have both enjoyed it, I believe.
Yeah. Well, I was born and raised there and was there for my first 2 decades. So not all in the same neighborhood, though.
Yeah. It's a good place to be.
Yeah, absolutely.
Well, on that note, let's see your presentation. Take it away.
Okay. See if I can share properly here. And what happened there? Yeah, lost the sharing. I promise this worked in our, there it goes. Okay, good, we're all set there. You can see.
Yep, looking good.
All right, perfect. Okay, so once again, thanks to both Kelly and Scott and to invite me to to participate in this. Hope it's a lot of fun. Please, put up any questions you might have. I'm going to, when we were first talking about what to discuss here, sort of threw out a couple ideas, and one was to talk a little bit about smell in the time of COVID, because that's certainly something that's become quite prevalent in the news and affecting a lot of people. And then we also talk about the idea of about odors are really the ability to give you the ability to communicate in a variety of ways. And so in the end, I decided to combine this a little bit. So we'll talk about both sort of briefly, hopefully link them together. But I think one concept that people don't often think about, but the smell is important for communication, and it's true across a variety of species. Even the National Institute of Health Institute that funds research into smell is the National Institute on Deafness and Other Communication Disorders. So smell is important for communication. We know about it. You see it all the time in animals that are sniffing each other to try to get information about each other. These are social cues, and we'll talk about those briefly. Predators can use odor as a way to find their meal. In this case, you have a mosquito using odors that are emitted from a human host to get that blood meal. Now, in the case of humans, we probably don't have innate responses to certain odors that what we think of as like pheromones, like some of these other animals might, but we certainly do use odors as a way of interacting with our world, whether it's with each other, a mother, the smell of her baby, an adult with their partner, just recognizing the change in the seasons, freshly cut grass, flowers, and things like this. Smell really connects us to the world around us. And as we were just talking about a moment ago, while smell and taste are certainly critical for helping us to make assessments about the palatability and the freshness of food and what we may want to ingest, just the very act of eating and drinking is really at the center of our social lives. So in this way, smell and taste are very critical aspects of how of the experiences where we communicate with each other, whether it's a holiday, a standard meal, out a night on the town after work, whatever it might be. And then smell can even give us critical information about our world that helps to keep us safe. Smoke or gas leak that will alert us to impending danger. Spoiled food can give us particular smells or taste that warns us about it. And animals will even be able to detect potential predators by the sense of smell, although this little guy here seems to be concentrating more on the smell of the food rather than his impending death. But what we will talk about here in just a few minutes are some of those types of social odor cues that go between species or among species that allow animals to make judgments about their environment and to make appropriate behavioral responses. But before we get to that, I mentioned at the beginning, we certainly have gotten, as a community, a much greater understanding and respect for the importance of smell in our daily lives ever since the COVID pandemic began. And I'm going to start with, this is a TikTok video via YouTube. It came up a little bit. I'm going to play this for just a moment. This gentleman posted after having COVID, this was in 2020, about his loss of his chemical senses. Okay, so you can see there that that guy actually had all three of his chemical senses affected. So sense of smell, which allows you to detect volatile compounds either in the air or coming from your mouth and going up through the back of your throat. Taste, which is primarily sweet, salty, sour, bitter, or umami, the savory taste of glutamate. And then chemesthesis, which is one you're all familiar with, even though you probably don't know the term, which is the ability to detect compounds like the capsaicin in a hot chili pepper, the coolness of menthol that's in mint, compounds in onions and garlic and such that elicit painful or temperature effects and sort of irritating effects, and actually the basis of the Physiology or Medicine Nobel Prize from this last year, or the discovery of the receptors for those. It was apparent very early on in the pandemic that smell and perhaps taste and chemesthesis were affected with COVID-19. And these are a couple of bits of data from a study. It was done by the Global Consortium for Chemosensory Research, which is a group of hundreds of researchers from around the world I was involved with as well, where we used sort of crowdsourced surveys and going in 28 different countries, 28 different languages, I should say, to get feedback about people's sense of smell, taste, and chemesthesis with COVID. And on the left here, we see the changes, we're just looking at the sense of smell here, for people who had been diagnosed with COVID, either via just a clinical assessment or a lab test, and the change of their ability to smell from before they had gotten COVID-19 or during their disease. As you can see by these dot plots and these density diagrams, most of these people had a pretty good sense of smell. They rated up near 100 or certainly above 75 before their disease. And then after COVID, a large number of them showed almost no sense of smell was present or vastly reduced. If on the right here, we look at a greater group of this same survey population, comparing people with or without COVID-19 this time, as opposed to people with COVID-19 before and after, we see that those with COVID-19 had reduced smell, reduced taste, and reduced chemesthesis. Although the chemesthesis loss was less common, as was the taste. Smell loss seemed to be, at least in the earlier stages with the alpha variant of the SARS-CoV-2 virus, affecting 50 to 70% of individuals who had symptomatic COVID-19. So it was prevalent. It was dramatically affecting the ability of people to use their sense of smell and sometimes taste and chemesthesis as well. And we knew for a long time, but even before COVID-19, that smell disorders, olfactory disorders, could be caused by a variety of different things, including viruses that lead to upper respiratory tract infections. Common coronaviruses, rhinoviruses, adenoviruses can lead to temporary or permanent smell loss, and the absence of the sense of smell we call anosmia. But there are also sinonasal disease. Some people born without the ability to smell, it's called congenital anosmia. Early stages, Parkinson's and Alzheimer's disease often has smell loss associated with it, and head injury. Traumatic brain injury very commonly can result in a olfactory loss. So this is really common. Smell disorders, even before COVID-19, were prevalent. One major study associated with the NHANES program found that over 12% of Americans 40 and older have an olfactory disorder. And these are just a list of some of the variants. We have some quantitative disorders, anosmia and hyposmia, either complete absence or partial loss of smell. Qualitative loss disorders, parosmia and phantosmia, either distorted smell or smells that are not there. It's basically olfactory hallucinations. And then age-related smell loss is very common. The elderly usually show a fairly pronounced loss in the ability to smell. So this is a common health problem. And as we talked about at the beginning, smell itself is more than just a convenience. It really impacts our diet, it impacts our health, it impacts our interactions with each other and our connections to the world. So hopefully one of the silver linings of the pandemic will be a greater appreciation and understanding of the implications of smell and taste loss. And we're already seeing an uptick in the research associated with assessing these disorders, and then hopefully also identifying cures, which at the moment we do not have. So we'll switch gears slightly to talking about smell more broadly, but I want to give you a little context here. So what do the chemical senses need to do? They need to detect all relevant chemicals in the environment. In some cases, they might be novel ones. In some cases, they may be ones that have co-evolved with the recipient of those odors and tastes in order to elicit a particular reaction. The system has to encode the quality. By quality, I mean sweet or sour or bitter, not good or bad, and the quantity of individual chemicals, and do so in the context of complex mixtures. Talk about that in a moment. And then all this has to be done in a way that the brain can decode this code, it can interpret the sensory information, and do so in the context of your experience, your motivations, other sensory inputs. An example I'll give is a number of years ago, there was this common product trend of making things clear. So one of these were crystal clear colas. So they looked like a sparkling water or a Sprite or something like this, but they were cola. And unfortunately, they didn't go over well, in part because the visual disconnect of, you say, oh, I'm going to drink a Sprite, and it tasting like a Pepsi, was just that disconnect in expectation from the reality of the flavor, what was disconcerting. That was partially part of the demise of that line of products. So we have to put all of our recognition of most smells and tastes in that experiential context to know that that particular odor means rotting fish, while that one means pepperoni pizza. We learn that. Some tastes, not odors, are innate in their response. Sugar is appetitive. It promotes ingestion for all of us. Bitter compounds or sour compounds, if given in isolation, are aversive. They tend to connote spoilage or poison. Although certainly for those of us like beer or coffee or endive, you can learn to appreciate bitterness in the context of known foods. But I also point your attention to this quote up here. Hay smells different to lovers and horses. So hay is just hay, and it gives off whatever odorants it gives off, a group of chemicals, an aroma, a bouquet. But what that particular smell means is very contextual. Horse is going to think of it as food. Lovers may be thinking of it a place to have a tryst. These are, but it's the same odor. What is different is the experiential context that people or animals put with it. As I mentioned, odors and tastes are almost always encountered in complex mixtures. This is a jasmine flower, and if one wanted to recapitulate the aroma of jasmine by just putting certain chemicals together in precise combination and relative quantities, you could get pretty close by including all these chemicals. One thing that the olfactory system, sense of smell, needs to do is to recognize all these chemicals in a mixture individually, but do so in a way where it can be put back together to make sense, so that you can make sense of it. And you can differentiate that it's a jasmine flower or a rose or asphalt. Of course, there are also some very specialized chemicals that serve as odors for animals and may carry some very specific type of information or elicit very specific behavioral or hormonal responses. And we collectively call these semiochemicals. So these are olfactory signals, some of which are volatile, some of which are peptides or proteins that can be detected via the nose. And they're for both either intra or interspecific communication. These are just examples of the major classes. So pheromones, you've probably heard that term before. A pheromone is a olfactory signal given off by one member of a species, detected by another member of that same species, that evokes a usually stereotyped behavior or hormonal response in the recipient animal. So in this case, this mouse gives off this odor cue, this one detects it, and then this one changes its behavior or something about its physiology. Pheromones benefit both animals, both the donor and the recipient of that odor. And a couple examples are, for example, male mice in their tears have a peptide that when detected in the nose of a female mouse will cause that female to exhibit mating behavior, specifically one called lordosis, which is a postural behavior that will promote mating. There are a variety of other types of compounds, some of which are volatile, some could be in urine and could be in the coat, other glandular secretions that can do that. Chiromones are given off by one animal, received by another, to the benefit of the recipient. And so you can think of sort of a predator warning signal in this case. There are compounds like one protein called FELD4 that's in that can be isolated from the fur of cats that will, when smelled by a mouse, induce freezing behavior or avoidance behaviors in this recipient mouse. A lot of carnivores, most carnivores, I think, have in their urine a compound PEA, phenylethylamine, that will elicit those same types of defensive behaviors. So chiromones are really warning and protect generally are protective for the recipient. Allomones are protective for the donor. For example, they can be, plants may be giving off compounds that will be aversive to predatory insects that would normally be feeding on them. And cinnamones are compounds that it's a little more complicated, but they're going to benefit both the donor and the recipient. So one example would be trees that are being predated by one type of insect give off a compound that attracts a parasitoid insect that will then use that predatory insect as a host for its eggs. So it ends up protecting the tree and also enhancing its own reproduction based on the tree-derived olfactory signal. So it benefits both. So these are all different types of semiochemicals that all elicit innate responses, so that the animal does not have to have experienced it before in order to, when it receives the signal, to have an appropriate behavioral response. But my lab over the years has worked a lot on a particular group of chemicals and responses that may be a little more sort of a blend between those semiochemicals and learned responses like we see with most odors. Before I get into that, just to briefly highlight how animals are detecting these is to take advantage of, okay, so I'm going to be just 2 more minutes. Don't want to run long, is to take advantage of olfactory subsystems. Basically, these animals have a variety of different groups of cells in different organs. This is true in mice, this is true in insects, that allows them to detect different chemicals. In this particular one, I'm going to give you an example of, if I can get it to advance. They're known as GCD positive olfactory sensory neurons because of the receptor they express. There's one called guanyl cyclase type D. We can label those cells with specific genetic markers so we can see where they are in the nose. We can see where they project in the brain. And what I wanted to illustrate to you just very briefly before wrapping up is the behavior that they underlie. And this is something that's called the socially transmitted food preference. If you are a restaurant, you're talking to someone else about, oh, this food's really good, or this is horrible, don't try it. We can use language and facial expressions to communicate that. Mice use odors. So the one animal eats a flavored food, another animal smells that and then has a choice. They will prefer the food that has the same flavor. And this is dependent on these particular neurons being intact. So if the neurons are intact, the animal forms a preference for that demonstrated odor. If they're not, they can't smell it. They do not form a preference. They do not care one way or the other. And what these odors are, they're peptides that are expressed in urine. There are volatile compounds, carbon disulfide, that are in breath, that when paired with the food odor, the recipient animal associates those and learns that they should prefer that food. And we think that that's a way of conveying that the food is safe to eat, although you can certainly override that. So I'm going to skip the last slide, which just shows a little bit more of that, and just highlight the people who did the work. Those in bold here are the ones who had a particularly great contribution to that work on those GCD neurons I showed, and collaborators in that work, the GCCR and principal collaborators there, and at the University of Florida for the work in the area of COVID-related smell loss and funding from the NIDCD. And thanks a lot for your attention, and happy to answer any questions.
So that was awesome. And I now have like 5 hours of smell and taste related questions that I'd like to ask you. But I'll note real quick to let other people in on the game that if you have a question, they can put it in the Q&A box, and then Scott and I will ask those questions. But okay, so I'm super excited that you started by talking about mice and how they respond to smells. So my day job is I study parasites and how they manipulate the behavior of their hosts. And so you probably know that I'm going to ask about Toxoplasma gondii. How about, can you explain what we know about how that impacts the way mice and rats smell and how they respond to cat urine? And
yeah. There's been a lot of debate about how that's impacting mouse, whether they're basically shifting their it's changing their innate aversion to the cat cues, and they're no longer able to detect those, whether it is actually flipping what we call the valence, how they feel about those, whether it's a positive or negative one. And I can't say that I've looked at the specifics of the literature in the last few months, but yeah, I think the jury is still out on exactly how it is changing the mouse's ability to properly respond to predator cues. And so that you can imagine these could be all sorts of different reasons, because they don't detect it, they missed it, they misinterpret it, or there's something completely else that's unrelated to their olfactory ability that is changing how they respond to fear, how they respond to motor programs, all these. And so it's still up in the air.
And if I remember, so every once in a while I jump into the Toxoplasma gondii literature, and it seems like there's not consensus on lots of things. Like sometimes you see it in mice, but sometimes you don't, and it's more in rats, or sometimes they specifically respond to bobcat urine, but not fox urine. So it seems like it's really specific with cats, but you don't always find that. Are the effects even consistent? Like, do you feel confident that this is a thing that's actually happening, or is this a thing that maybe we've been tricked into thinking is happening?
Well, I think I'll give you actually one concern you probably don't need. So a lot people may not recognize, excuse me. People will not recognize how where we often get all these urines. We don't go out and have a pet bobcat or a pet fox. There's a company called Predator P that sells a lot of these urines that get used in some of these types of studies. Problem with sometimes using urines is certain of the components can degrade over time. So one possibility is that that the stimulus is varying from. So it's not only the species of origin of the stimulus, but the quality of the stimulus, how well it's been preserved and all the compounds that are present in it. And if we look at cats, there are compounds in their urine that are clearly predator signals to rodents. And if we look at, but they also have compounds in their fur and other ones. So it's a complex, there's not just one chemical that's going to be doing it. So the animals, the recipient animals, making a very complex judgment in that respect.
Got it. And I see that Scott helped us all out by putting a link to predatorpeastore.com. So I'm sure
excellent.
it's made it
Welcome.
easier for everyone. Thank goodness.
And obviously a lot of their business is to sell it for people to put around gardens and stuff to keep animals, deer away and things like that.
Got it.
Yeah.
So they're not necessarily concerned with what researchers like you
I think it's part of it, but it's not. I don't think that's the primary business. Yeah.
Okay. Thanks.
Mm-hmm.
I have to say, you totally took me back with the references to the clear colas like Crystal Pepsi or whatever it was. I hadn't thought about that in a long time. So thank you for that. But no, that was such a, I like Kelly, I wrote down like all these questions. I've got like 1,000 questions in my mind, but I'll try to stay focused. So one thing I did want to ask you about is, what is your take on human pheromones? Like that's like been this controversial idea for a long time. Like, where do you come down on the idea that, do humans have pheromones?
Yeah. So in most cases, what the compounds that we think of acting as pheromones in a variety of animals work via a particular portion of the olfactory system known as the vomeronasal organ. And this organ is present up until, well, I know that there may be some vestigial aspects of it, but sort of at New World monkeys, when they become sort of the shift between dichromatic and trichromatic vision, is about the time evolutionarily when the vomeronasal organ and the vomeronasal receptors for these compounds disappear. Linear. So old word apes, humans don't have a vomeronasal organ. So right away, that's one aspect that is missing. But at the end of the day, our main olfactory system could conceivably detect compounds that we have co-evolved to recognize and have an innate reaction to. To my knowledge, that no one has demonstrated a convincing example, no matter what the back of the magazines say about, if we still have magazines, that about how that the one that sort of led the charge for many years was the idea of having synchronized menstrual cycles, or menstrual cycles that would shift with smelling t-shirts. And I think that's even more debated now, to what extent that is truly an olfactory related phenomena. So yeah, I would lean on the no side, I guess, would be the shorter answer.
Mm-hmm.
Yeah.
Do they debate that happens at all, or they debate that it's olfactory linked?
Both. I mean, my understanding of a lot of it may have just been a sort of a sampling bias. If you get enough women in one location, you're probably going to find people on a similar schedule.
Yeah, fair enough. What about that smell, that t-shirt smelling, where you pick the people who smell the most different than you? Does that hold up?
So yeah, so that's with major histocompatibility types, and certainly in rodents, that is something that can happen. So rodents can detect differences in MHC haplotype, and actually a rodent that has mated with animal of one detects a different MHC haplotype from another male, will spontaneously abort the litter they have, sort of the idea that they're going to prepare for the male that's around. With humans, there's some evidence that you can recognize odors associated with those genetic differences. And we all do have differences in the odors that we give off, and how much that is related to that particular aspect of our genetics or not is still unclear. But yeah, rodents can recognize diseased animals, other animals with it, and they give off a specific, which is sort of the basis of people trying to develop sensors or use dogs to detect different diseases, cancers and such. And there's some people that seem to have sensitivity to that, even though a lot of the compounds haven't been identified.
Simon, did you want to chime in with a question, or should I go ahead with another one?
Yeah, I also have so many questions. So I feel always weird about this because I'm not like a biologist that studies another organism. I study humans. And so I'm always interested in like what the experience is like of having this thing, which you can ask the human about, sort of, but you can't ask a rodent. And so this is like not a scientific question because you can't answer it scientifically. But like when you think about what it's like, what these rodents must be experiencing with this like more intense communication through smell than maybe we have as humans. How do you think that experience of the world is different?
Yeah, I mean, it is a hard question to answer. I mean, and you will see arguments all the time. It's like, well, the dog has more, millions more olfactory receptors than do humans, and therefore they smell better. They're better at smelling, and that's probably actually not true. For if you look at the research that is able to measure sensitivity to different types of odors, humans are actually quite good at detecting odors. We lack, as I said, that vomeronasal organ that allows us access to a lot of these social cues, a lot of these semiochemicals. That is different. So it's hard to relate that. But the big difference, I think you're sort of alluding to, is how much weight you give that sensory input versus vision or audition or stuff. We are certainly much more visual creatures and auditory creatures than just what we're doing now. We are looking at each other. We are listening to each other. We're not smelling each other, but we're able to communicate really well. And rodents would probably not do Zoom very effectively. And so, but then again, how much is it, how plastic is that? How much could that vary if you weren't able to take advantage of one of those senses and had to focus on others? Is that capacity still there, and is the capacity still able to adjust? But yeah, we have to with rodents ask the question in different ways, and they may find, technically they don't find things sweet. That's a perceptual content. They find things appetitive. We can measure whether they like it or not. We can measure that they like something equivalently to sugar, something we find sweet, but actually calling it sweet is a bit of a anthropomorphic naming of it.
Fascinating. I feel like we could probably go on for quite a while. I certainly have a bunch more questions, but we probably should move on so we have time for our next guest. But thank you so much, Steven. That was absolutely fascinating. And if you're able to stick around, we will at the end try to come back and have a few questions that are for both you and Simon.
Great.
Thank you very much. So our next guest is Dr. Simon Fischer-Baum. And Simon is a colleague of ours at Rice University. He is a faculty member in the Department of Psychological Sciences at Rice University. And Simon's height of his fame, his peak of his fame happened at the age of 1. And when he shared this with us, it blew my mind. This is incredible. Simon appeared at age 1 on Sesame Street. And we actually have a clip here that Simon has shared with us of his cameo here on Sesame Street. So let me share my screen and give you Simon Fischer-Baum at age 1 on Sesame Street.
This one's the one that's me.
That's you, Simon, right? Not
Yeah.
the first kid. So these are babies learning to walk, baby people and baby animals.
You were clearly destined for greatness from the beginning, Simon.
I was really hot stuff in my childcare. They would like every time I was on Sesame Street, they would like yell and all the kids would gather. It's like the only thing I remember from being like 3. There's our like super 80s car. It's all there.
How did this come about? Were you like, did your parents like send in a video? Like what was the, like, how did this happen?
Well, we had a family friend who was made short films for Sesame Street, and she had a daughter who was like maybe like 3 months older than me, and she had this idea for making a movie, but the window for being able to make this movie with a kid is extremely narrow. And so by the time she got it together, her kid was no longer appropriate. And so she just like shopped around for who she knew that had like a kid of the right age. And so me and that other person who's like also a meaner like me were the people she knew. Oops. And there's an ad.
Wow. Amazing. Fantastic. Well, all right. So that may have been the height of Simon's fame on TV, but now he'll be famous once again presenting here at D.O.R.K.S. So Simon is going to talk to us about the many facets of language in the brain. All right, take it away.
All right, here we go. Okay. Yeah, so this is, yet another YouTube clip for me to be able to for people to watch. People at Colleen's house can get really excited about this one too. Okay, so I'm going to talk about language in the brain, and I'm going to talk about, some of it's going to be about the work that I do in my lab here at Rice, but mostly it's just going to try to provide an overview for thinking about language and how our brains are sort of organized around language, and actually how complex language is as a system to make sense of. And so I want to start, I guess I'm making fun of some of my colleagues that are like 2 floors up from me, but I have some really excellent engineering and scientist colleagues at Rice who got a big grant from DARPA to build this new technology that's called Moana. I almost certainly because they both have or all the people on the team have like small children, though they like swear that it doesn't have anything to do with the Disney trademark that they're infringing at this moment. But they are building a device and it's very cool. It has like a bunch of like optical imaging set up and a bunch of magnetic stimulators. And it's going to be this cap that you can wear where you can take a thought that exists in one person's head, decode it with a computer, and then like write it and put that thought into another person's head. And it, they're doing really cool stuff, and the technology is really fascinating. And I think they'll make some progress at being able to do this to some extent. But as their neurobiology of language colleague a couple of floors down, I always laugh when I hear about this, because in fact, as humans, we already have a really good system for how we take an idea that exists in one of our heads and figure out how to put it in somebody else's head. And even if you like show the whole image that my colleague put together for his proposal, he even knows that this exists, which is language. Language is the system that we have where we have an idea inside of our head, some mental image, some idea that we want to convey, and then we have a way of transmitting it to another individual so that they can get the exact same idea in their head. And while it's an imperfect system, it's going to be better for a long time than the Moana system that the neuroengineers at Rice are developing. And so when we think about language, we really want to think about it. I like to think about it in this way, right? I like to think about it as a system for taking ideas, of doing this type of conveyance of ideas from one body to another body. And it goes through a bunch of different systems, right? So it is something that exists externally in like sound waves that are moving through the air. It interacts with our various sensory organs. So it's something that has to come in through our ear if we're talking about spoken language, or our eye. So if we're talking about sign language, it's our written languages. And then it interacts with our motor system, either our like mouth and tongues as articulators, or hands, depending on the type of language that we're speaking. And then there's this whole, and this image, which is from this great online exhibit about language at the Yale University Library that Claire Bauern has put together. In here, in very small, much smaller than it takes up in the whole space, is the brain, right? So you can get the signal in and out, but a lot of what has to happen is making sense of the signal and figuring out what signal it is that you want to produce. Now, it's an imperfect system, right? Language is filled with ambiguity. And so we often find ourselves in circumstances in which the idea that's in one of our heads that we're trying to convey, we convey it imperfectly, and we get hilarious hijinks around people misunderstanding each other. And so a classic example, this one's from Michael Pitts at Reed, is the ambiguous sentence, "the chicken is ready for eating," where you might be thinking about is the chicken the one that's doing the eating, or am I the one that's doing the eating of the chicken? And so we have sentences that are ambiguous, and so it's not always getting that mental image from me correct into the mental image into your head, but we're doing our best as we're doing it. So language it's not a, I don't think technology is the right word for it, for at least for some aspects of language, but it is the system that humans have, the best system that humans have for this process of taking, like what we're doing right now, taking the ideas that are in my head and trying to put them into your head. Of course, there are limitations of language as a mind reading technology. So some are these inherent ambiguities that we have. There's other issues that come up, which is that sound only travels so far, but we've, as humans, have developed technological innovations that allow us to overcome this, like the telephone or the internet, the Zoom screen. All of these things are allowing sound to travel farther than what it would naturally do. The other thing is that the signal is ephemeral. So once I speak, that signal disappears. But we've also developed technologies that allow us to address this limitation. Like if you're watching this on YouTube, the signal of my speech has been made permanent and can be watched at any point in time. Those are both sort of the technologies that I'm describing are kind of like 20th century technologies, maybe late 19th century technologies for how we address these issues of language, the limitations of language as a mind reading technology. But of course, probably, I don't know, maybe it's just because I study written language. I think probably the greatest human invention, the greatest technological innovation that humans have ever made is the creation of written language. So the creation of written language shown here all the way on one side of the screen with the Rosetta Stone allowed us to be able to communicate the sort of language signal permanently with permanent marks, so it's no longer ephemeral, and allows it to travel as far as we want it to. You can put it down on a piece of paper or a heavy rock and carry it with you wherever you want to go, so that the exact things that were communicated stay in a way that lots of people can then receive the message, even if they're not in the same place or at the same time. So language is something that exists inside of the brain, right? So our language is both something that exists in the world. It's these marks or these vibrations of air, these recordings in ones and zeros or on some type of analog tape. These are all sort of aspects of like an external version of language. But for us to be able to create this language, we really have to think about the systems that exist inside of our brain that allow us to be able to produce and understand the language that exists in the world. And so as neuroscientists, we are a big fan of like cracking down on pop neuroscience that exists in the world. And so one of the like main things that people like to think about the brain that we like to say that's not really true is that there's some people who are left brain people and some people that are right brain people. That's like something that I don't quite know where it came from. It's kind of like we only use 10% of our brain. It's like a myth that exists and I don't really know what it means. And we all are both hemisphere type people. But that's not to say that there's not differences between what our left and our right hemispheres are capable of doing. And so for almost everybody listening, certainly if you're a right-handed person, I can be extremely confident that I would be able to find the language stuff of your brain inside your brain's left hemisphere. If you're not a right-handed person, if you're a left-handed person, I would still be pretty confident. I think it's around 75% of people who are left-handed who also have left hemisphere language. But for some people, language is on the right-hand side. And the reason I know this largely, or the reason we know this largely, comes from the fact that when you have damage to the left hemisphere of the brain caused by all sorts of things, but in a lot of the work caused by stroke, so a hemorrhage or an occlusion of one of your cerebral arteries on your left-hand side, you're often left with a pretty severe impairment, at least initially, in your ability to speak or understand language, or read or write or do something linguistically. Whereas if you have damage on the right-hand side, it's usually not something that leads you to have a language impairment, or at least not a really overt one. There's interesting subtleties about what right hemisphere stuff might be doing when it comes to language that I'm really happy to answer questions about if you have them later on. Okay, so when you have damage to the left hemisphere language network, and really, it's super common with many, many different left hemisphere strokes. You have something that sort of is clinically called aphasia, which is a language disability caused by brain injury, difficulty speaking, difficulty understanding, difficulty reading, or difficulty writing. And there's a lot of work that's out there to just try to make people be more aware of aphasia. And so when I thought about this as like a public talk, one of the things I just wanted to make sure of was to just make people aware of aphasia. And if you feel like you don't quite know what that is, you would be kind of with the majority of the country. So most people, 85% of people in like a recent survey of Americans had never heard the term aphasia, had no idea what it was. The people who had heard it frequently had to do with the fact that they had somebody in their family who was suffering from aphasia. But at the same time, it's really common. And so it's something that you should know about when you think about the people that exist in the world around you. So more people have aphasia than things that you almost certainly know about: cerebral palsy, multiple sclerosis, Parkinson's, muscular dystrophy. Aphasia, about 250,000 new people a year end up having aphasia because there's just a lot of strokes that occur every year, and about a third of all strokes result in aphasia. And for many people who have aphasia, within a year, they've kind of recovered their language abilities, so they don't notice the impairment anymore. But there's also a lot of people where the aphasia that they have is chronic. And so they end up sort of with a long and lasting difficulty with some aspect of language. So there's an estimated about 2 million people in the US who are living with aphasia right now. And I'm talking about it in terms of stroke, but another really common cause of aphasia are like variants of dementia that aren't Alzheimer's disease, but like the cells that are dying as a result of degeneration are sort of, it's happening in language areas of the left hemisphere. And so people are left with like what is called primary progressive aphasia, where like it's a dementia where the primary symptom is a language disability, and because it's a dementia, it's a progressive disease. You can also get aphasia because of tumors or traumatic brain injury, all those kind of same things that like can give you all sorts of other disorders of the mind that happen as your brain gets damaged. But so I think, there's this tension in the work that I do about just like generally raising awareness around aphasia, because a lot of the work that we do in the lab is with people who have aphasia. I have some brains that you can sort of see. We have people who've come into the lab who've had strokes, and as a result of their strokes, they have language loss, and we study what that language loss looks like. It's hard to see, but we 3D print their brains after we scan them. And so you can see kind of like where different people have damage. This person had like a right middle cerebral artery, a left middle cerebral artery infarct, which left like this whole chunk of cortex in here lost. This person had one that was the posterior cerebral artery. And so you can see like the back of her brain had like a big hole in it, right? And so we want to talk about aphasia because aphasia is something that people should be aware of. And like these people who all have language loss, they should all sort of, there should be an awareness of what's happened to them. And oftentimes, oftentimes people, they sort of, this happens to them and they feel like the way that the world engages with them really changes. And so it's useful to talk about it as a single thing, but it's not really a single thing. And the reason it's not really a single thing is that language is really a lot of different things that are put together. And so knowing language means knowing a lot of different things. And so what I have here is just like a dictionary entry for the word lexicon, which is like the fancy jargony word that we use to mean dictionary, right? So when you look at a dictionary entry, you see that there's lots of different kinds of knowledge about this word that are all here, right? So there's knowledge about the word spelling. You can look up the word spelling, its pronunciation, its syntactic category, it's part of speech, its meaning, what we call it semantics, also its etymology, where it came from. And not all the stuff that's in a dictionary we think exists inside of the head of a human that understands language. Like probably as you learn the word lexicon, you're not like thinking about how it originally came from Greek and then it has shifted to modern Latin. Like that's probably not a type of language that you know. But the rest of this stuff, or some variation on the rest of the stuff, is all things that you know when you know language. And so as we think about how language exists in the brain and how brain damage can cause damage to the language system, can cause aphasia, we have to think about how there's different types of knowledge that are encoded in the brain about language, and how damage to any of these locations that are encoding these different types of knowledge can lead to something that is aphasia, a loss of the ability to speak or understand or read or write. But it doesn't mean that two people can have the exact same problem when it comes to aphasia. And so here's just some examples of some kind of cases that show really striking differences between different systems of knowledge. And so here's a patient whose syntax, so ability to kind of speak grammatically, is intact, but like the words themselves are not intact, right? And so when asked to write a description of this picture, the patient writes, "The box has been negated by the misused synex." Right, so the grammar stuff, the parts of speech, the structure is all there, but the words themselves are not. This is just this patient being asked to fill in sentences that are heard. Just a minute ago, we turked everything. She drank 4 of the Yallards. The man is being dress whacked. Each of those things are sort of nonsense, but they're not nonsense that's coming from nowhere. They're nonsense where the structure is all there, but the words themselves are not the right words. And so this is one type of aphasia that you can have, where you get the grammar right, but you miss the meanings of the words or the words themselves. And this contrasts with, say, another patient where they're asked to describe this picture. This is a really common picture that you have people describe as you're trying to assess what kind of aphasia they have. It's like a whole mess of a bunch of disasters about to happen. And so this is a patient just describing what's going on. And it goes, "So this is a boy Will fell accident. I mean, it seems that, what is the name? I don't know, but crooked in the chair, and this too. Oh boy, it seems like get cakes to give to go. Not sure. Let's see. I'm not sure what this is. That's a water in the sink. The water is full. The water down to floors, and also I guess the floors is wet there, and then like a woman." So in fact, when you look at this passage, some of the words are slightly mispronounced, but all the words are there. The description of the picture is actually quite accurate. The patient sees the picture, understands what's going on, is able to find the right words to describe it, but the grammar is all lost. The sentences are not going together fluidly, and you see things that are like frankly ungrammatical, like calling this like multiple floors here, or using the floors is wet there, right? Like there's things that are just not happening in a grammatical sense. And so that's a dissociation. These two patients both have aphasia, but they're super different from each other. And so in fact, when we look at aphasia going across all sorts of different languages, all sorts of different patients, we'll see that there's different kinds of aphasias, and they roughly correspond to different locations of the lesion, different places in the brain that there's been damage. And because of that, there's different types of language impairment. So we have patients who are diagnosed as having anomia, which is difficulty in naming words. Another set of patients might have what's called pure word deafness, where they can hear things, but not words, but they can understand words, but they can speak, and they can read, and they can write just fine. So it's just about mapping from sounds onto spoken language that they have trouble with. There's patients who, as a result of the stroke, have dyslexia or alexia, which is a difficulty in reading words, agrammatism, which is a difficulty with sentence structure, dysgraphia, agraphia, which is just like fancy Greek words for difficulty in writing words, right? So you see, as you start to look at somebody who comes into the lab who has aphasia, there's actually like any number of things that could be disordered about the language system, and two people could have totally complementary sets of skills and difficulties. which makes, say, figuring out what the treatment is for aphasia or the cause of aphasia something that is like an ill-formed question. But even these labels are like overly general, because even if you dive deep into the literature and the kinds of patients that come into the lab who have one of these labels, you see that there's enormous amount of variability in what they're actually having trouble with. And so I'm just going to focus on alexia. A lot of the work that we do in my lab is focused on written language disorders. And so these are the kinds of written language disorders, reading problems that people have when they show up in the lab, some of whom I've sort of worked with myself, some of whom I've, I have colleagues that have described these cases, right? And so some people come in, so these are people who have trouble, all these people have trouble reading words. They come into the lab, you give them words to read, they struggle. All right, but why are they struggling, right? So there's some people who have what's called aperceptive alexia. And so these people, when they come in, their problem is that they can't even see the letters, or I'm going to show you a case where it's actually the numbers that this person couldn't even see. But it's not a general visual problem. They can see other types of visual objects. And oftentimes it's not even knowledge about letters themselves, because when they're asked to write the letters, they're actually totally fine at being able to do that. And so it's a perceptual problem, but a perceptual problem that's specific to the visual aspects of written language. There's other people who can see those letters, but can't map them onto words. This is something that's called pure alexia. And so these patients, they are kind of like the pure word deaf patients, where the problem is not with the visual system itself or with the language system, but it's being able to map from vision onto language. And so they can often write words still. There are other types of language knowledge is totally fine. Sometimes when you like hand them like letters to feel with like a tactile, like refrigerator magnets, they suddenly can understand what the word is that you're spelling to them, but they just can't see the words to understand them. Other patients can see the letters, but they see them out of order. This is something that's called letter position dyslexia. Some patients can see the letters, see their order, but can only read them out loud if they're words that they've experienced before. And they can't pronounce unfamiliar words like the word flope, which sounds like a really esoteric kind of like test that doesn't really affect you in the real world, until you realize that that's true of a lot of names that they're experiencing too. So they really struggle in making sense of things that they've never experienced before. They can only find the things that they've memorized in some part of their brain or other. Other patients actually have lost the things that they've memorized, right? So they're totally good at reading these non-words, and they're totally good at reading words as long as their pronunciation is like completely guessable from the phonics. But as soon as you give them a word with a strange spelling like yacht, they start to really struggle, and they pronounce it like, I don't know, yatched, and they're unable to get to those pronunciations. And finally, you even get these patients where like they see these words, they know what they mean, but they get to the wrong word. And when they get to the wrong word, the errors that they're making are they're saying something that's semantically related. So you show the word cat, and they read it aloud as the word dog. In each case, a different part of this pretty complex system that we need for even a single word reading, for going from like a visual input all the way through to a spoken output, has gone wrong. And there's many steps along the way, and damage to any of the steps along this way can lead you to have this kind of problem. And these steps are located in different locations in the cortex, such that you can have a stroke that impacts one, but not the rest of them. So I'm just going to show you one of these cases just to give you a sense about how precise these deficits are. So I picked one that's like been published in the literature. So like if you're curious about it, you can go look it up. It's a PNAS paper that came out 2 years ago now that I think is really good. This was a patient that actually I met him, and I knew him a little bit when I was in graduate school. It took them a long time to write up the case, but he's somebody that they were testing in Baltimore when I was in graduate school. And so this person didn't have a stroke. This person actually had a neurodegenerative disease that one day he woke up and he wasn't able to see numbers anymore. And it was really it was really a struggle for him. He was a working engineer who was doing like a lot of like diagram building and like had to do a lot of calculations. And he was like going to see clients, and he could no longer see the numbers. Particularly, he couldn't actually name a visually presented digit. Like if you gave him a visually presented digit between 2 and 9, 0 and 1 for some reason, we're totally fine with him. But between 2 and 9, he just couldn't name them at all. But he had no problem in other types of visual tasks. He had no problem seeing letters. He had no problem seeing other types of visual objects. line drawings, faces, things like that. He had no trouble seeing mathematical operators, plus, minus, percent sign, times, divide. Those things were all fine. And he actually didn't have any problem with his mathematical abilities either. So as long as he could get the numbers into the system, so if he heard the numbers or read them as whole words like 5, written out FIVE, or Roman numerals like IV, meaning 4, like all of a sudden then he was able to process those numbers, knew all the stuff about the numbers, and was able to do all the arithmetic for them. But just to show you sort of like, I can describe these results, but I want to just show you the results of an unbelievably simple task that they did with him, which is just copy what you see, right? And so he's shown this 8, and his task is only to just draw the thing that's showing up in front of him. And this is what he drew. And anecdotally, I don't think this makes it into the paper, he was only handed an orange marker. But while he was doing this copy, he reached across the table to grab a black marker to be able to, oops, I raised my hand by accident. He reached across the table to grab a black marker and started to draw in here. And so there's tons of variation. I just saw Scott's note, and so I'm going to finish up in like the next 2 minutes. I'm sorry about that. Okay, so language is really a complex and interactive system, and it involves engaging much of the brain's left hemisphere. And so different nodes in the language network have different jobs, and damage to any of these nodes or the connections between these nodes lead to an impairment of language, thus aphasia. But depending on what the node is that's damaged, the kind of language impairment that happens is going to be different. That's one source of variation. The other source of variation is that for some people, the brain is able to recover and reorganize after damage. And so there have been these reports of people who've had like a whole hemisphere removed, especially when they're children. The left hemisphere gets removed and suddenly they're able to do language totally fine just using their right hemisphere. And so we see that there's actually something that's going on also about reorganization that we truly don't understand, which is a big question going forward. So who's going to show brain reorganization and recovery and who's not? How are these sort of different factors of age, social support, the type of lesion, the type of impairment, access to treatment impacting how people's brains are reorganizing? All right, so now here's my sum up. So just as the take-home message to sort of drive home the big points that I was trying to make today. So language is really important. It's a huge part of what it means to be human, but it's not a thing. It's actually many different interconnected things for us to be able to produce and understand language. These things reside in different parts of the brain, mostly in the left hemisphere, and damage to the system causes aphasia, which is both super common and also extremely isolating because language is such a core way of how we engage in the world. But because language is so interconnected and there's so many interconnected things, aphasia is super varied. And so people in this audience are not clinicians and they're not doing work with people with aphasia, probably. And maybe if you are, then that's awesome. But if you aren't, you might know somebody with aphasia and you might know multiple people with aphasia. And I think the take-home message really is then, how do you make sure that you treat each person as an individual and don't just assume something about them because you've seen somebody else with the same diagnosis. And so with that, I put up a slide of all the people that helped me do all sorts of things in my lab. And thank you so much. It's fun to give a talk here. Sorry that I went long.
No problem. That was awesome. Gosh, I always feel like I have way more questions than we have time for, sadly. But so Simon, I'll probably come track you down and start asking you questions in your office one of these days. But one thing that you said that I was kind of fascinated by is you mentioned 3D printing the brains of patients that you're working with. That like you mentioned it somewhat in passing, I think, but like I was kind of stuck on that idea. Is that something that anyone can do? Like, how do I go and get a 3D model of my own brain? And what kind of information would I be able to get from that if I don't necessarily have like a brain injury? Would it be meaningless, or could that be like the next 23andMe, like where we all have 3D models of our own brain sitting on our...
not at the, it's like mostly vanity, I think, at this point. I mean, I think for the patient, it's not meaningless. So you need to go to like get an MRI and get like a really good structural MRI scan. And so we do it at like, a type of imaging that goes beyond what like is your run of the mill MRI. So it's mostly like if you're going to like a regular clinic, it'll be like a 1.5 Tesla magnet, and we use like a 3 Tesla magnet. So it's a slightly better image. But then we really relied on our like neuroengineering friends upstairs who what happened was they participated in some of our studies, and then they were like, oh, we're just gonna go 3D print our brain. And we were like, oh, we can do that? How do we do that? And so we started just doing it with like the people in the lab for fun. But I noticed that a lot of the participants that were coming in who had strokes had so it's really bifurcated. So some people really don't want it. Some people like really don't want to think about like the big hole that's inside of their head, that it's really upsetting for, I think, good reason. But for other people, it's like if you like look at their phone, like the front of their phone, their like home screen is like a picture of their scan. And like it was those people that we were sort of first like, hey, would you be interested in seeing like a 3D printing of your brain? And they were like, yeah, we would totally be interested in it. So we started, because we see people repeatedly, see a small number of people for a long period of time, we started like giving them out as like, thank you for participating in our study if you want them. But from like a scientific perspective, it's just so different. Like brains, like when you do human neuroimaging, you're almost always looking at like a bunch of two-dimensional images, and you're trying to infer a three-dimensional image from that. It's different than like the physicality, I think, of like cutting open like a mouse brain and like actually taking it out of the body, which I think you do if you're doing like animal neuroscience. The way that my understanding of where the damage was in these patients changed as a function of just having a 3D model in front of me was really striking. And it's made me realize that it's, I think, important, even though it's like the same information, it's important to be able to have that it in 3D.
I can tell you from having taught about medical students neuroanatomy a lot over the years, is that one of the biggest challenges is, because they're generally seeing things in atlases and images like that, just translating those two-dimensional pictures into that three-dimensional space, which technology is starting to let us do, but it was always one of the biggest challenges for them.
Yeah.
So I have a comment that I've been dying to make, and I'm going to try to like find the question as I go, so I'm not like that guy. But so when you were showing Moana, it made me think of the brain-computer interface people that I interviewed while writing Soonish. And I promise we get through lots of Dorks episodes without me mentioning Soonish, but this one happens to be sort of Soonish heavy. But so one thing that surprised me that they kept saying was that like an end goal of brain-computer interfaces is that we'll all be able to like immediately share all of our thoughts with everyone. And it seems to me that like a function of language is to create certain kinds of relationships with people, like nice relationships with people. And that gets harder if you can't not say some things that you're thinking. Because like I feel like that's an important part of the relationships we have with people, is like keeping some stuff inside. And so to try to like ask a question, is that ever part of aphasia? Like, do you lose your filter and you say things you don't mean? And maybe the brain computer interface people should just be trying to get us to do more of that with, I don't know, targeted brain strokes.
Yeah, I mean, I like watched the technology develop with horror as somebody who's like both a neuroscientist but also a social scientist, as like Facebook invests heavily in no longer having you like type your messages but just think your messages. It's like yeah. So I mean, I'll say a couple of things. So one thing is like most of these, one thing that's really striking about aphasia is like you lose language but you don't lose you, right? So the ability to like to build up messages, to understand something that's going on in the world, the ability to like have this rich, complex, oops, something's about to ring, sorry. This rich, complex tapestry of like your identity is still there. It's just the ability to get it out. So I think you're right that like language is really good at like filtering. And there are people who like lose the filter. And so there's like both like dementia types and also like aphasia types, where like the outcome is, we call it a loss of inhibitory control, which just frequently, it means like a lot of very subtle things when we're talking about like the tests that we give in the lab. But what it also means is like warning the people that are going to test that person that this person might say some really off-putting things to you, and like it's because of their stroke, and like I don't really know what else to say about it. But yeah, it seems like language as a filter, language not just as like a, like anything in my head should go in your head, but as a filter is another good way to think about it.
We have a question from Jessica Alexander, who's asking, is there any work/evidence of aphasias and even higher level linguistic connections than the ones you discussed, such as pragmatics, like that is mapping a message to an intent?
Yeah, so we're interested in that. I mean, I'm mostly like a word nerd person, so we tend to look at individual words, but I'm like shifting up, like Wordle is like my, you know, nirvana of like scientific thinking. But we are shifting up. I have people in the lab who are really interested in shifting up. And so we are interested in this thing called discourse processing, which is like your ability to tell a story maybe, and your ability to like, you know, think about things as having a beginning and a middle and an end. And you definitely see that's true, that there are patients who as a result of brain damage, that's where they end up having lost things. And it's a kind of unfortunate situation because the tests that you give to determine if clinically somebody has aphasia mostly doesn't measure that. And so these are people that are like sort of like tested by a speech pathologist and told, oh no, you're fine. Like all these measures that we have, you're fine on them, but they have trouble returning to work because like they can't, like there's aspect, and they know that they have these difficulties going forward. And so we're trying to figure out what those difficulties are, but those tend to be understudied. And then there's the right hemisphere stuff, right? So the right hemisphere stuff is a lot of where this stuff happens. So right hemisphere strokes to language areas will often affect kind of like paralinguistic stuff that's just next to language. And so sometimes it's like your ability to detect tone. And so patients, like when you look at like marital discord between somebody who's had a right hemisphere language stroke and their caregiving partner, it's like the problem is like the person doesn't understand sarcasm or doesn't understand urgency. Like they understand all the words, but they don't understand like how the tone is being carried in those words to convey extra information. And there's growing evidence that that kind of thing is happening in sort of these right hemisphere regions. So it's not that the right hemisphere isn't involved in language, it's just not as, it's a subtler involvement.
Sounds like the way that communication happens online.
Yeah,
like,
right.
You can miss, there's no tone, and so people miss sarcasm or other
yeah, exactly. Yeah,
nuances.
It turns out that like prosody is important, like the way that we say things is really important.
Right. So, okay, I have a question that this might sound like a silly question, but I'm actually curious about whether there's any basis for this. So I'm curious about learning multiple languages. So like, you know, I learned Spanish in high school and in college, and then later I learned Portuguese. And my sense was that when I learned Portuguese, it sort of replaced my Spanish to a certain extent. And I always have this sort of like cartoon in my mind that like there's a place in the brain for like other language, like foreign language, and that, you know, there's only room for one in my brain. And I'm well aware that there are many people out there that speak 5, 6, 7 or more languages, and I'm in awe of them. But for me, I feel like I can do English and I got room for like one other one, and it's hard to maintain more than one. Is there any neurological basis for that, or is that just on me?
I mean, there's plenty of, so that some of that probably has to do with the fact that like you learned English and like that was like the language that you was spoken in your home and in your community, and then like you had a separate experience that taught you Spanish later, and it's not, and like you're not immersed. Like I know that you have spent time in like Brazil being like immersed in like a Portuguese speaking environment, but it's not like it's not like your ability to communicate with people fully depends on your Portuguese. And so I think that's a common experience, but for people who are like learning languages as not their primary means of communication. I mean, like my grandmother's first language was Yiddish. She doesn't speak any Yiddish anymore. She now only speaks English. I don't think she's spoken Yiddish for like 85 or 90 years even. But like it's not like she could learn another language. She could learn French, and it's not competing with her English at that point because English becomes this thing. But there is, I mean, there's a lot of interest in trying to figure out like the bilingual brain, and especially like bilingual aphasias. But because everybody's brain is so different, or I'm sorry, everybody's stroke is so different and language is so complex in the brain, it's really hard. And also everybody's experience with languages is so different. It's really hard to try to draw conclusions about what bilingual aphasia is, or what, or sort of how that's situated in the brain. So like different parts, like words, maybe there's like two separate lexicons for like words, but like overlap for like the sounds of the language or something like that. It's hard to know how it works. There are sort of interesting things that are sort of general observations, which is when you lose language because of aphasia, it seems to be more likely that you lose your second language as opposed to your first language. And this can even happen in cases like my grandmother. So it didn't happen to my grandmother, but it did happen to a patient that we were testing in Baltimore who was like, spoke German until he was like 8. And then he like came to the US and he like spent his whole life in the US and got married to like somebody who only spoke English. And he just like, all his language was English. And then he had a stroke and he could only speak German. And like his English was fully gone. His German was impaired, but it wasn't as impaired. And the lab was just lucky to have like a German RA, or sorry, a German postdoc at that point who could like talk to him, because like otherwise there was like nobody that he could communicate with in his family. Nobody else in his family spoke German. And so there are super interesting things that happen when you think about, that must mean that there's some separate storage like you're sort of imagining.
So one thing that's interesting to me is, so you both study brains, and like brains are an important tie-in for both of what you do. And so I studied brain infecting parasites, and I started getting into the brain stuff. And at that point I was like, no way, this is way too complicated. I have no idea what's going on here. I'm just going to focus on the parasites. So I guess I'm wondering, like, how do you two tackle such complicated questions? Like, what is the right mindset? And also, how did you two end up getting into these fields in the first place? Maybe we'll start with Steven. Oh, I think you're muted.
Yeah, sorry. Yeah,
Okay.
I was muted myself. There we go. To answer the second question first, it was when I was an undergraduate, I had interest in neuroscience and in marine biology. And back in the days there was a Xerox piece of paper that went around with all the professors and what they worked on that gets spread around and say, oh, here's someone working on reflexes in mantis shrimp. That sounds interesting. That's this reflex arc and such. So I went and said, oh, well, actually, we're not doing that. We're working on sense of smell and crayfish. So any interest in that? It's like, okay. And so 30 some years later, I'm not doing crayfish anymore, but still doing smell. As far as sort of getting at that, all the complexity, I mean, certainly in the olfactory system and the taste system, there's a lot of distribution of how information is represented. The further up in the chain you go into the cortical regions of the brain, they're dealing with these chemical senses. And frankly, in many ways, I'm much happier in the nose or on the tongue, where those detectors, where that information is first getting encoded and starting to be sent back before it starts to be processed, not only within its own bucket of smell or taste, but then starts to be put together with all of these other types of information and the memories and experiences that the animal will have using for the context of that. So part of the way I deal with it is to not deal with it.
I also
That was my strategy as well.
So I'll answer this. I guess I'll like follow up on that. So I also feel like I deal with it by not dealing with it, which is I'm showing images of brains, like I'm showing these 3D printed of brains, and it's like important information for me, but it's not like the ultimate goal of like my work, right? So I didn't like provide in my like talk any like, this is part of the brain that does this function, or like this is the mechanism by which it does this function. I could have, but to me, like knowing exactly where something is in the brain, which is like what a lot of cognitive neuroscience questions are, is kind of uninteresting. Like it is there somewhere. And being able to get at like a more precise, like, what is the precise neural code for how this works is also something that like, once you get to these higher order things, it's like unclear how you do. So I feel like I want to keep a pin in it for complexity though, right? So there's a lot of neuroscientists who are trying to attack it from like Steve does. So from the like, let's start with the things that are really tractable and understandable, where we can really actually understand the receptors and understand what they're responding to and turn them on and off and get like, a real understanding of what's going on. And I just want to keep in mind that that is really far from what it means to experience the thing and what we think of as what it means to have thoughts. And so I'm interested in higher order cognition, which is language and memory and other kinds of things, because I'm really interested in just remembering, like being able to define how complex that problem's going to have to be for you to be able to go from this sort of low level, what is a neuron responding to this like much more complex system. And so I'm totally comfortable with never having a satisfying biological answer to any of my questions. I'm happy to like exist in the world of thinking about mental processes and how those must divide somehow by the system. And then how I got into it was I was really interested in, some of it was I read like Oliver Sacks books and I was like, this is fascinating. And then I got to college and it was like, oh, this is a job. When I was like, there was a lab at my university where somebody was doing work like this. And I was like, I could like do this. And that was very exciting. I also was really interested in neuroscience. I was a neuroscience undergrad, but I think even at an undergrad, aware that like science is very rewarding sometimes. And sometimes you feel like you're doing really like things that are making the world better, but it's not all the time, and it's not even most of the time. And so I was sort of weighing that in my head as I was thinking about what I wanted to do. And I thought, like, I could do that and have like a bad science day and then come home and be like, also like I slaughtered 50 mice as a part of my like scientific study. Or I could be like, I like hung out in this home of this old person who had a stroke and I like spent time with them and I changed a light bulb. And like, that was sort of like, to me, that kind of like trade-off of like the human side, the like hangout part of the human work was really valuable to me. And so that's what pulled me over. And I liked language in part because there is no animal model. And so like, or the animal models are like potentially questionable, right? People do songbird work, but it's not obvious that songbirds are doing with their Broca's area the same thing that what humans are doing with their Broca's area. And so I was excited to work in an area where I wouldn't feel pulled into doing animal work.
I love that. And you know, I love this combination of both of these talks. I mean, both of you have such fascinating insights into, you know, into the human mind and the human experience. And I'm wondering if you could each maybe tell us a little bit about what do you think are the limits to the extent to which we may be able to someday use technology to imitate that human experience in your own field. So like, you know, as we're moving towards, you know, developing artificial intelligence, how far can we take that in terms of, say, language for Simon, and in terms of sensory perception, like taste and smell for Steven?
I go first. Do you want me to?
I can go first.
Okay, for it.
So, I think whoever named it artificial intelligence was like a marketing genius who also like missold the entire thing. So we have systems that are capable of transcribing human speech pretty well, not perfectly. We have systems that are capable of writing stories based on like in the same style. I don't think we have anything that actually understands. Like, I don't know what it would mean to have an artificial intelligence that's able to create a mental model that's like equivalent to like what we're actually doing with language. Like, we can do some of the peripheral operations to a better or lesser extent. And like, we can be congratulatory on like the fact that like it gets the easy stuff right 50% of the time. And like the harder stuff we can just like ignore. But I think that we're really quite far. And I don't even really see what the path would be towards creating something that's like using language really in the way that humans use language.
Yes, I would put it maybe a little more broadly in technology and say it's a bit of a bi-directional thing. One is certainly taking the knowledge we're learning about the way taste or smell works and trying to use those neural networks, use those receptors. There are different ways that people are taking advantage of them to create sensors that can be valuable in a variety of contexts, whether it's sensing disease. You go in, someone waves a wand over you, they get the volatiles that you're putting out and they say, oh, we think you may have cancer, we need to do additional testing. Putting them in agricultural settings. The corn is ready to harvest. let's get that, because certain volatiles are coming out. Those are things that are happening. Those are already. But then the flip side of it is using some of that information to create prosthetics for, especially for people who have anosmia or other type of smell or taste disorder, where they're having difficulty in actually detecting that chemical world around them. The challenge is, of course, is that you want to make sure that you're not basically providing nonsense or providing unpleasant information. I mentioned these qualitative disorders of parosmia and phantasmia, where you're either having distorted smell or phantom smells. Those in many ways can be much more disruptive and disturbing for people than the loss of smell. Having parosmia, the distorted smell, which a lot of people with sort of in long COVID are experiencing. They can detect odors again, but now smelling a flower smells like feces or like burning rubber, or food is just, especially certain triggers like coffee and chocolate and others, can really cause these absolutely horrible, disconcerting perceptions. You don't want to install something in somebody's nose to detect it, and what they're getting is garbage smell all the time, as opposed to the things that they want to detect. So it's a challenge. I would say we're a long way away from being able to do that, but people are starting to think about those possibilities.
I also think, just to give Scott a chance to plug his book, since Kelly plugged her book, I think that when we think about these assistive technologies, like, we don't need to think about humans as being like a fixed form. So as one might read in The Future of Humans, that humans are evolving in a context of the technologies around them. And so one of the reasons I study written language is, like, that's a technology that humans actually have had access to for thousands of years, but it's a technology that was invented. And there's like pretty good evidence that, I mean, it certainly is the case that, like, our brains, once you learn how to read, are kind of wired to learn how to read. And it would be interesting to think about whether within those sort of 3,000 years, that's been enough time for our brains to sort of have evolved in some ways to be kind of like attuned to literacy. And so as technologies develop to be assistive in these ways, our brains are going to adapt to them as well.
And I would highly recommend that everybody get Scott's book and read Scott's book. Scott, was there anything that you wanted to say after Simon's comment?
I mean, there's like 1,000 things that I want to say still to both Simon and Steven. I had a bunch of questions about evolutionary history in the context of both language and taste and smell and our future. We'll have to save it for another time, but thank you to both of you. This was just absolutely fascinating. My mind is filled with a bunch of ideas and questions, and probably going to get some follow-up emails from me. But
yeah, this was really fun.
Yeah, this was great. Thanks so much for having us.
Yeah, thanks so much, guys. This was awesome. And for anybody who is who's tuning in now, we'll put this video up on YouTube if you want to watch it again in case you missed anything. And you can keep up with us at D.O.R.K.S chat on Twitter. And yeah, we are working on scheduling additional events, and we'll announce those on our Twitter account when we get those locked in. So yeah, thanks again, guys. This was super fun. I had a blast. I'm going to go have dinner. Dinner tonight is Ukrainian themed, and I'm going to make everybody listen to smell and brain facts the entire time. They're going to be kind of tired of me by the end, I think, but I had a bunch of fun. So this is all I'm going to be talking about, I think, for the rest of the night. So thanks, guys.
Bye.
Yeah, take care.
Bye.